Okay, well, it's wonderful to see all of you today virtually, and I am going to. This is day 4 of our NAN Metabolomics Workshop, and today the topic is fraction libraries. Yesterday we had SAND with Frank Delaglio. Today's fraction libraries, and tomorrow is what some information on C13 NMR for metabolomics. So without further ado, this is, you've seen this slide before in my introduction to NMR talk from day 2, and this is so this is a sample of cotton leaves that's resuspended in DMSO D6. You actually saw this same sample, the SAND analysis of this same sample yesterday from Chris. And so this we just illustrate this as a very complicated NMR spectrum where there's a lot of overlapping peaks and asking the general question, how do we deal with all of this complexity? And there are several ways of doing that. We've alluded to, but we haven't really spent a lot of time talking about 2D NMR, general 2D NMR methods. There's a lot out there right now that you can find. We use 2D NMR quite a lot in our lab, especially Colmar database matching, where you've seen references to this a few different times in my talks. You heard about SAND yesterday, and if you missed that, I would refer you back to day 3 and Frank Delaglio's talk on NMR data processing and SAND in particular, which is a time domain analysis to try to extract individual peaks out of a complicated NMR spectrum. The C13 is what we're going to hear about tomorrow, and today the focus is going to be on fraction library. But I do want to stress that combinations of all of these things above are routinely used in our lab. So it's not just a standalone. So we can use 2D NMR with fraction library. We regularly SAND fraction library. Sometimes we can do it on C13 NMR too. So we have, these are all tools to solve complicated problems. So the thing I'm going to be introducing is relatively new in our lab. I don't think it's widely used in NMR metabolomics right now. That's part of the reason I wanted to present it in this workshop, because we're finding it extremely valuable right now. But the concept is not new at all. The concept is really borrowed from natural products chemistry, where natural products chemists for decades have used a technique called activity-guided fractionation to find the activity that they're interested in and purify it in natural products discovery. So I don't indicate this here, but roughly 50% of all of the drugs that we take to treat human disease are derived from natural products that originally were isolated with methods similar to what I'm going to describe to you right now. This is what I'm just highlighting in this one slide is really just a summary of a gigantic amount of work and a whole other lecture but it's a study that we published a long time ago now in 2008, where we used activity-guided fractionation to purify a mating pheromone in the nematodes Caenorhabditis elegans. We did this with Paul Sternberg and Frank Schroeder's lab when my lab was at the University of Florida. So the general idea, I'm walking you through this just to show what activity-guided fractionation is and to give you a sense about how you can use this in your metabolomics applications. So in this case, we were interested in the fact that we could take material that was produced by hermaphroditic worms, put it onto a plate with no fractionation, and attract males to that spot. So in this, the assay is sort of summarized here, where where you would put a spot of of material that you're interested in scoring, material that's a blank, and then you can put male worms onto this plate and then track to see which spot they go to. So it's a relatively unbiased way of seeing what the male response to some material that you put onto the plate is. In our case, we were looking for something that would attract males to the hermaphroditic material that would have a pheromone for mating attraction. So we could do that. We were able to get male attraction from material that hermaphrodites released, and then we were able to test that material over different developmental stages, and we had a profile that would attract males according to when they started to become mature and able to sexually reproduce. So it all made sense. And then essentially what we did was we took our our material, in this case shown on this panel here, well, we control with no worms or with young adult hermaphrodites, and then we were able to purify this material. So in this case, we did very simple fractionation using solid phase extraction, and we cut it into three parts. Something that we could elute from a C18 solid phase extraction with 50% methanol, 90% methanol, or just collect the flow through. So we just had those three fractions initially, and you can see all of the activity that we were interested with where it was concentrated in the 50% methanol. So at that point forward, that's all we looked at. We didn't pursue the 90% or flow through at all. We got rid of a lot of compounds in that process. And then we basically, using the same kind of approach, we started to purify this 50% methanol and test it for activity with other types of separations, and a lot of ion exchange, for example. And we tested every single fraction that we were able to obtain, and things got more complicated after the initial stop, but we were able to follow fractions that had activity of males were attracted to the material. So the long and the short of this is that after we go through all of those steps, before we do any NMR measurement or mass spectrometry measurement, we were able to have a relatively pure sample of material that did what we were interested in it doing, and then we were able to identify mating pheromones from that. So this is just meant to illustrate what activity-guided fractionation looks like and the power that it can have. And around that time, we recognized that that the process that I just described in natural products chemistry, where you'll start with an extract, and you have some bioassay. In the case that I just went over, are male worms attracted to whatever fraction we have? You go through several different fractionation steps, and you end up with some product that is more or less pure, and you try to figure out what that is using NMR spectroscopy and mass spectrometry. That's the protocol that's used routinely in natural products chemistry. In metabolomics, by contrast, all of you are aware, the general concept is that you have different phenotypes that you're trying to compare. There could be, in the case of the cotton leaves, for example, that we've talked about, cotton that is more drought tolerant or not, or you might be looking for other comparisons, one genotype versus another genotype, some population of people who are sick and people who are not sick. And so whatever it is, metabolomics is trying to take a couple of different groups, do a statistical comparison between the groups, and then end up with a mixture of either NMR or mass spectrometry. And then what we hope in this case, so I'm highlighting there's an activity over here on the natural product side and an activity over here that one of those two groups has as an example. And then we can identify that activity in a statistical sense that ends up being the same thing that we're interested in activity-guided fractionation. So there are differences for sure between metabolomics and natural products analysis, but there are also a lot of similarities. And so the motivation behind the work that we're telling you about today is that this fractionation process has a lot of value and could probably be used a lot more often in metabolomics. We're not alone. So the Jeremy Nicholson's group with Matt Lewis as the lead author on this one a few years ago published something very comparable to what I'm going to tell you about, where they took a pool of human urine, did HPLC fractionation, made a fraction bank, and then basically tried to look for biomarkers that they were interested in based on the fact that they had already separated all this material. So it's a little bit different flavor than what I'm describing, but I recommend that you look at this reference if you want to see the approach that Nicholson's lab have used to try to get additional information for identification of unknown metabolites from this fractionation type approach. So now I'm going to transition into telling you how we are doing this in our lab, and you're gonna see later on in one of the interactive sessions, Chris, who many of you saw yesterday in a breakout room. Chris is going to be describing how to process the fraction data, and then you're also going to have John Grimes describe to you how we collect this under automation by NMR. But the general concept was also started by Gonzalo Gouveia, who was a graduate student in my lab and is now, and also Brianna Garcia. She did the mass spec part of this, but Gonzalo did the NMR part. Gonzalo is actually up in Cornell now as a research scientist with Frank Schroeder. But the work that I'm describing is between Chris and Gonzalo largely. So we start with the concept is really simple. There's nothing fancy here. You have a sample that you're interested in analyzing, and this can be some sort of a reference sample of what you're studying, or it could be a pooled sample from your study, an internal pooled sample, which we often will make in a metabolomics run. But the general idea is that the sample that you're inputting, you have plenty of material, and it's representative of the study that you're trying to look at. I put down here at the bottom a reference from a paper that Gonzalo published while he was in my lab on defining a long-term reference material that I think many of you might find useful. I don't have time to go over the details of it, but you might look at that reference if you want to think about how to make a reference material for your own study. So the idea is that sample is put into semi-preparative HPLC. We either use reverse phase C18 type chromatography or Hillock chromatography, fairly big columns. We're not trying to get perfect separation out of this. We're trying to just be efficient. And we just collect a chromatogram. The key here, and in our case, we can also do, we have a very low resolution single quad mass spectrometer for a detector in addition to UV detection. There's actually a video that we have on our public YouTube video that Chris made showing this process. So he'll refer to that, I'm sure, but that's up there right now on the Edisonomics video site, the YouTube site. So what we do is we basically collect a full gradient and then collect a 140, because it's the limitation and it's very convenient size for us, the limitation of our fraction collector. So we can collect 140 fractions. And the thing that's important for you to understand is that these are untargeted. So they're just timed fractions with whatever timing fits right in the gradient. And so, so there's no targeting. We're not looking at a peak and purifying the peak in what I'm about to show you. So this is an example of NMR of a region, a small region, shown on the bottom. So that the x-axis is NMR chemical shift, and the y-axis is the chromatographic separation. So this is time on the total ion chromatogram on the vertical axis, and showing what Chris was able to do with a sample from Neurospora crassa using Hillick chromatography in this case. And so the what you want to focus on is on the back, the mixture, the material on the back. Let me see if I can move this thing. Um, the material on the back is the unfractionated sample, the NMR spectrum of the unfractionated sample, and then the fractions are going down here below. So the key point and the reason why this is relevant to what we're trying to show in this workshop in general, if you focus, for example, on this peak, or you can pick a lot of these different peaks, but this is a particularly good one. So you look at something like that on the black, the black peak back here, or collection of peaks, and that looks pretty daunting. And we could deconvolute all of these peaks by sand, and Chris has done that. And in fact, he has this whole fraction library sanded as well. I think that we're looking at the Fourier transform version of it here. But you look at that peak, and you really have no idea without doing 2D or without some other method to know what's going on behind that. But what you see in the fractions, I think, are pretty amazing. That here's a species, here's a species, here's a species. There's something else going on back here. Here's something else. None of these things would be apparent all the way down as individual molecules had Chris not separated this by Hillick chromatography in this case. So it really is eye-opening to us to realize just how much overlap we are dealing with in NMR. And I have to say that I think that this is one of the problems in NMR metabolomics. We oftentimes sort of boast about the fact that we don't need much in the way of sample preparation in contrast to what LCMS needs to do for sample preparation. But I think it's to our peril that we're not doing more chromatography and cleaning things up. It's not to say that all of this information is not, we can separate. We can do 2D NMR and start to resolve some of these things, and people do that. So it's not like we're totally missing all of this, but just if you look at 1D NMR, you have to realize what the complexity is underneath that. So in Chris's interaction, or his demo coming up shortly, and then also in his breakout room, he's going to be showing you methods that he is working on right now. This is active work in our lab to try to figure out how to annotate these fractions. So one thing, for example, well, let me just reinforce the fact that we have these all in separate tubes. So these are all separate material of fractions that we have after we do that HPLC. Each one of those tubes we can also sample by high resolution LC-MS. We can collect, fragmentation data. We can go to databases like GMPS for LC work. We can do 2D NMR on any of these fractions that look interesting. We can, using the 2D approaches that have been really perfected by natural products chemists, you can come up with de novo ID, or you can match databases if compounds are in databases like Colmar. Fractions can be further purified. So these things are definitely not pure fractions. They're mixtures, but if we're interested in really pursuing one particular fraction of interest, we can go through and purify it further and actually isolate peaks and such. Within a fraction library, and I think Chris is going to show you a little bit about this, he's developing methods to use the Stocksy-like approaches, and I'm not explaining Stocksy right now. Chris will show it in more detail in his presentation, or in the hands-on this afternoon. This is statistical total correlation spectroscopy developed by Jeremy Nicholson's lab. And it's a very powerful way of putting together molecules, but he's been able to start to apply this nicely on fractions in a fraction library. You can also use any kind of one-dimensional database matching approach. People like Canomics, sometimes a commercial software. Colmar has a 1D data query. There are a lot of different approaches that you can use to just take a single fraction and try to match it to databases. So this is what you're going to hear more of from Chris this afternoon. But the thing that I want to also stress during this presentation is that fractions are also physical material. It's not just the NMR spectra that we have that you saw in that pretty figure that Chris made of Neurospora. You can do things with these things, and this is where I want you to think back to the natural products chemistry. You could do, if you're interested in a particular activity, maybe the organism you're working on has known antimicrobial activity. Well, you can test each one of those fractions for that antimicrobial phenotype, and then only focus on the ones that kill the bacteria that you're interested in. Maybe you're looking for pheromones, and if you put a fraction down on a plate, you can attract a worm, or you can do some other. You have to have an assay to ask the question if they're acting as a pheromone or antimicrobial. Maybe if this is an organism that would then be supporting growth for another organism, like, for example, marine phytoplankton, and release material to support the growth of marine bacteria. Maybe you could see what in that phytoplankton is a substrate for bacterial growth, and the list goes on. Whatever biological question you want to ask, you could do that. You could come up with some sort of a test that would become your assay, and you could start to test these fractions. And then you can also do things like, you could say, do these fractions bind to macromolecules, proteins, RNA, et cetera, other macromolecules? And that's what I'm going to focus on for the rest of this talk, is what you could do with this macromolecular binding. So the data that I'm going to show you are in the next few slides are related to a project that the Cannon Lab in the biochemistry department at University of Georgia, they study a really interesting enzyme called fructosamine 3 kinase, FN3K. This is a small molecule kinase that has it's been understudied compared to a lot of other kinases. Structures are known. In some cases, they just know that the substrates for this kinase are small molecules, and Cannon's lab are trying to study details of this. They came to us because they had evidence that FN3Ks would bind to various NAD species, and they wanted to see if we could find experimental evidence to demonstrate that. So now I'm going to just tell you about some of the techniques that we can use to study these things. This is work in progress right now in our lab, but it's fairly interesting and straightforward. And I think it's important for you to be able to know about these kind of things. So saturation transfer difference is a technique. STD NMR is a technique that's used routinely by the pharmaceutical industry for screening targets of interest for binding. And usually if a company is working on a protein that they want to drug, then they will have a large library of molecules, small molecules, that they can then screen using a technique like STD or other related NMR techniques that I'll mention at the end, to see if things bind or not. It requires weak to moderate binding, but it doesn't work. It's not appropriate if you have tight binding or really strong binding ligands. Requires a large macromolecule. In fact, the larger the better. So big proteins work great. There's really no limitation on the size other than solubility. And the ligand is usually 20 to 100 times more concentrated than the macromolecule. So we're using a roughly millimolar ligand concentrations, and then I think that in these experiments that I'm going to show is around 25 times less protein. And the general concept is this, shown on the right, and this is from a review article here, an old review article that's great from Jeff Peng's group, showing how this concept works. So there's some generic protein of interest, in this case FN3K, and you have a mixture of different ligands in your solution represented by the stars and circles. In this case, the circle will bind to the protein, but the star doesn't. and it has a characteristic on and off rate. And actually, it's the on and off rate that count more than the KD, but usually that translates to moderate to weak binding. So the idea is that if you saturate the protein, and when I say saturate, what I really mean is that you remember from my introduction to NMR, I talked about the two energy levels, alpha and beta, that a spin 1/2 system has in a magnetic field. Well, saturating is making those two levels equal to each other, and that's a non-equilibrium state. But you can do that by putting in RF saturation that hits the protein, but it doesn't hit the pool of ligands. And if you do that and just keep that protein saturated through a process called spin diffusion, the saturation will transfer through the protein very efficiently and then jump onto the ligand, which is in the active site. That ligand then goes off and through various binding events going on and off and on and off, it will enrich saturated ligands out in solution that are binding to the protein. Okay, so that's the general concept. What you do in practice is perform two experiments. One is off-resonance saturation or a reference spectrum. So you go way far away from the protein of interest and put in a saturation pulse. And then the next one, you go to the protein of interest, put in a reference pulse, and you do these back to back at the same time or in the same series of experiments. In one case, you get a spectrum that say looks like this, and on the top, and then the other case, the saturated compounds, the ones that are binding to the protein, are diminished in their size. If you take the difference of these two, then you see a peak that shows up positive as the compounds that bind to that protein. So it's very powerful. It's used for lead screening a lot in pharmaceutical industry. And here are some data that Conrad was able to get with one of Cannon's postdocs on this project. And basically, he looked at several different species of FN3K, from people to plants to other mutants and stuff that I don't even know what it is. But they tested all of the different NAD species, and these are all positive binders over here and here, not so much when they make a mutant that they knew would obliterate the binding of these NAD species to that mutant. So that shows pretty clearly that these are binding. And now the reason I'm telling you about this here in reference to the fraction libraries is that Conrad was also studying at the time Caenorhabditis elegans, and he had available to him a mixture of C. elegans metabolite, polar metabolite. So the top one is saturation transfer difference spectrum as the reference, where there's no saturation done. The center one in green has saturation so Conrad put FN3K into this mixture of C. elegans metabolites, the same FN3K that he was just looking at on the last slide. Then here it's on resonance, and then the bottom one is the STD subtracted spectrum. And lo and behold, he was able to see these NAD-like species showing up out of all of the worm metabolome, a little bit of other stuff too that I'll tell you about in a second, that binds to this protein. And so this is now the fraction library that Conrad made with C. elegans. This is also helic fractionation. And this is the complete fraction library, complete mess, hard to see. There's just so much stuff in there. It's just hard to get an idea about it. But he's highlighted a few sections in here where you can see different fractions that do bind. This is actually NAD plus. There's another place where we have NAD plus, and so he's starting to analyze and identify some of the species that are in here that bind. And now, just for the last couple of slides that I'm going to go through here before I turn it over to John Grimes, I'm going to show you different regions of this very complex spectrum to just give you an idea of the richness of the information that you can get from this sort of experiment. So on the left is part of this warm fraction library. On the right is the same fraction library screened with the FN3K added to each fraction. I didn't make that clear before. So there's this fraction library on the left. It doesn't have any protein. On the right, it has FN3K added to it in a saturation transfer difference experiment done. So you can see, for example, there's a big binding peak in here, corresponds to here. The stuff next to it that show up here they don't bind. The things up here maybe a little weakly, up here they don't. So you can just look and by inspection recognize what regions are binding to this protein. And a lot of these are not FAD species, or NAD species. So we realize that this is a way to unravel things that we just didn't know about. Here's just some other regions. I'll just show you a few more. The right is STD and the left is just a pure fraction library, just to give you an idea of the sort of information that you can get by thinking in terms of a fraction library. So a lot of information, a lot of things potentially binding, and for us, this opens up a whole new area of functional metabolomics is the term that we're using for this. So the binding data do need to be verified because saturation transfer can result from non-specific interactions. And so other types of binding measurements with synthetic compounds is important to validate that this is actually real binding. And there are other NMR methods that we also have going in our lab that are very complementary to STD. For example, there's something called water log-Z the reference is right here. This is a really nice way to screen an alternative to STD, or we've been running them with STD now. T1-Rho is another experiment that a reference for that is shown here, applying this to protein metabolite interactions in E. coli. So this is the last slide. In summary, basically, we think that metabolite fraction libraries are really useful. We're using them a lot these days. They can reduce spectral overlap in NMR spectra, similar to the way you could do that with 2D NMR. Repeated injections can concentrate samples. So if you have a very small amount of material but you have plenty of it, I mean, a small amount of sample, but a large volume, you can just continue to do auto injection and concentration and increase the concentration of the small peak. Compounds can be annotated with several different approaches, either with natural product chemistry type approaches or database matching or a mixture of everything. Isolated fractions can be tested for functional activities such as protein binding, like I showed, or other functions.